All right, so I've got my information on the board. Some of it's even legible. And like I said, the plan for today is to finish a little bit of information about T-cell effector function that I did not get a chance to talk about last time. The information that I have put on the board is actually information. I've got two little kind of table-ish things here, and they are tables that both are tables I think that you've seen in past lectures. And so we're going to talk about those. Then we will switch gears and talk about this paper by Bura et al. Remember that you have some questions due on that paper, so I am happy to take those. Also, the keys to both the T-cell development and MHC problem sets are posted on Moodle, and I have all of those problem sets graded for you. I have the grades in my Excel spreadsheet. I did not type them into Moodle yet, but I will do so. I still need to get you the grading back on the PRRs, but that one doesn't have as obvious like of an answer key that's going to help you studying for the exam. So that's why I did not prioritize getting that one back to you at this point. A couple people on one of the problem sets turned in problem sets that were missing, like scans of their problem sets that were missing a page. So if you happen to be one of those people, email me the missing page and you can get the rest of the points back. And I think I sent you an email as well, and like in the middle of your problem set it says missing page. So just be aware of that. Remember that we are going to have a question answer based review session tonight at 7 on Zoom. So please bring your questions. Also remember that for the exam, which is on Friday, of course, at our next class meeting, you can have an index card. And so if you need an index card, I've got some here. Last time we were talking about CD4 T-cell subsets. And I've drawn on the board the table that we were making as we talked about different CD4 T-cell subsets. And so in every case, when we were talking about these CD4 T-cell subsets, basically what we were thinking about is this idea of our naive T-cell getting activated by some antigen presenting cell, receiving some polarizing cytokine, turning on a transcription factor, and that inducing some kind of effector cytokines that have a particular role. And so our T-cells all start out as CD4 naive T-cells, but can get pushed in these different directions. And I've listed the details of Th1, Th2, Th17, and T follicular helper cells that we talked about last time on the board. The piece that I did not get a chance to tell you about last time is a lot of detail about the fifth and final helper T-cell subset that I want to tell you about. And so we need to think about this final helper T-cell subset. So the final helper T-cell subset that we didn't have time to talk through last time is known as the regulatory T-cell, or the Treg subset. And with the Treg subset, we have so what I'm going to first tell you is that we have a really important transcription factor that allows a Treg to be a Treg. And that transcription factor is called FOXP3. And you can see FOXP3 here on the slide in a couple of different places. When a naive T-cell starts making FOXP3, it becomes a regulatory T-cell. And the key cytokine that is made by regulatory T-cells is IL-10. And that IL-10 that is made by those regulatory T-cells will allow those regulatory T-cells to turn off or inhibit other types of immune responses. And you can see that happening in the figure on the upper right, and you can also see it just sort of listed in this table at the bottom. I'm going to skip ahead for a second and just show you a few different ways that regulatory T cells or Tregs are able to inhibit other types of responses. Sometimes it is that they can directly inhibit those cells. Like we can directly inhibit the cytotoxicity of different cell types, or directly inhibit dendritic cells and tell them to stop presenting, or directly inhibit other cells from producing cytokines. When regulatory T cells were first described, before we saw that those regulatory T cells had FOXP3, one other thing that we knew about them is that they have a ton of IL-2 receptor, especially the high affinity IL-2 receptor. So we actually used to distinguish them based on their high amount of IL-2 receptor, which had its pros and its cons. But we also know that these regulatory T cells can basically soak up all the IL-2 and kind of steal the IL-2 so that there isn't any left for other cells. And so some of this might be about inhibiting other cells using that cytokine IL-10. And we think there are probably actually some cell surface proteins as well, but that's a little controversial. And some of this is about being a cytokine sink and just stealing all the IL-2. But all of these are ways that Tregs can inhibit other types of immune responses. And broadly, I would say Tregs can inhibit, this is showing them inhibiting T cells, they're inhibiting antigen-presenting cells. This doesn't show them inhibiting B cells, but we think they can. They can kind of inhibit anybody else that you want here. What you will notice that I have not put onto our little table is how we polarize Tregs, how we make a cell a Treg. And the reason why I didn't tell you that is because Tregs are a little different than the other helper T cell subsets because there are two options for making a Treg. There are two different ways that a cell can be made into a Treg. And there are terms for the two different kinds of Tregs. I don't really care that we know the exact specific terms. I care that we kind of get that there are two different ways that we can make a Treg. One way that we can make a Treg looks pretty similar to what we've seen with these other subsets. And so one way that you can make a Treg is that you can activate a naive T cell in the presence of the correct polarizing cytokine. And so in that way, it looks very similar to what you've seen before. The polarizing cytokine in this case is called TGF-beta. And it is a cytokine that can also be thought of in a lot of cases as a sort of suppressive cytokine. And so if I go back to my original Treg slide, you can see we're polarizing those Tregs with some TGF-beta, turning on FOXP3, and making some IL-10. If you remember when I was telling you about the helper T cell subsets, I told you that some of the cytokines we can be a little squishy with, but I cared to use some stuff about TGF-beta. So now notice that we see TGF-beta in our table twice. We see TGF-beta both with the Treg, and we see TGF-beta with the Th17. The difference is that Th17s need TGF-beta plus inflammation. This is one of our classic inflammatory cytokines. While Tregs just have TGF-beta. And so if we're in a situation where TGF-beta is made, then we should turn off immune responses. We should be regulating immune responses. This is actually going to be one of our peripheral tolerance mechanisms that we'll see going forward. If, however, there's some inflammation around, the cell is not going to turn into a Treg. It's instead going to be a Th17 cell. What you might also notice is this could potentially mess up. If we were supposed to be making Tregs because we're making TGF-beta, but something goes wrong and we have inflammation at the same time, we could screw things up and get Th17s when we meant to get Tregs. And so that distinction of TGF-beta versus TGF-beta and inflammation is an important distinction. And so this is one way that we can take a naive CD4 T cell in the periphery and turn it into a Treg. And I'll say for our purposes, I might call that making Tregs in the periphery. We can also, however, have some Tregs come out of the thymus as Tregs. So in all the other cases, the cell comes out of the thymus as just a plain, undeclared CD4 T cell, and then it declares its major based on which polarizing cytokine it gets. But some Tregs come out of the thymus already polarized as Tregs. And when we talked about T cell development in the thymus, we talked a lot about 3 different outcomes that could happen. And at one point I said to you, and there's a 4th outcome, and we'll deal with that later. So if you remember, when we talked about T cell development in the thymus, we talked about what would happen if we had no or very low affinity for that T cell, no pats on the head, and that cell would undergo death by neglect or would fail positive selection. We saw cells that would have very high affinity, and those cells would get a very strong signal, and those cells would die by negative selection. And then we sort of have this medium category. And I told you there's really kind of low medium and high medium. The cells that get the low medium, here I've got it as low intermediate affinity, are likely getting some signals from MHCs around the thymus, but not really, much coming from the peptides. Those are the cells that are going to be positively selected and are going to become a T cell. As that signal gets higher, however, that cell gets kind of riskier and riskier because it's getting more and more signal from self-antigen. Not necessarily getting enough signal that we're like, I definitely need to kill this cell, but that cell is a little bit sketch. And so if we're in a situation where that cell is getting high intermediate affinity in the thymus, that cell becomes a Treg. And and you can see that here, this image doesn't show no affinity. So it doesn't show death by neglect. It just says the low one gives you the T cell, the high one gives you apoptosis, and sort of in the middle of those two, we turn on FOXP3. It's the same transcription factor. So it's still, stuff happens, we turn on FOXP3, and the cell is a Treg and can make IL-10 and do its Treg stuff. But here, it wasn't that we saw TGF-beta in the periphery, it was that we got that high intermediate signal in the thymus. And you can see this in this figure as well. So you can see that some CD4 T cells will come out of the thymus as Tregs if they had high, sort of high intermediate affinity for self-antigen. And so that's the one place where Tregs are a little different than our other T cell subsets. I have 2 other slides to mention. One of them is just a summary of the helper T cell subsets, similar to our table that we drew on the board. But the other thing that I wanted to mention about the helper T cell subsets as we're talking about them is that if you remember back to thinking about Th1 and Th2 cells, back in the day when life was simple and we only had Th1 and Th2 cells, we would often talk about the ways that Th1 and Th2 cells would regulate each other. I told you about some of the feedback between them. That not only does TBET turn on Th1 stuff, it also inhibits Th2 stuff. And not only does GATA3 turn on Th2 stuff, it also inhibits Th1 stuff. So I told you about this sort of cross-feedback or cross-regulation between them. Now that we've seen that this is more complicated, we have more T cell subsets, the question becomes, well, what goes on with all of the regulation here? How does regulation happen when we have more than just the two? And the answer is that all of the subsets regulate each other. So pushing T cells down any one of these paths also inhibits the other paths. So here you can see Th2 is inhibiting this Th1 or is inhibiting this Th17. And you can see Tregs are inhibiting everybody else. And so you can see that it's the same kind of thing, that each subset further gives positive feedback to its own subset's production, as well as turning off or inhibiting production of some of those other subsets. And so kind of once the response gets started down one path, we have more and more production on that same path. So that was the information that I wanted to tell you about T cell subsets. After the exam, we'll talk about B cell subsets, and we'll start talking about kinetics and memory and all that good stuff. But today, now for the rest of the time, I want to talk about the paper that you guys read.